you on Planetterrian Daily, episode 109 for July 3rd, 2026. Let's get into today's research and health developments. UCLA researchers identified a protein that slows muscle repair in older stem cells while also extending their survival under aging stress. UCLA scientists found that the protein NDRG1 accumulates in aged muscle stem cells and acts as a brake on activation after injury. The same protein improves cell survival during aging stresses, creating a trade-off between repair speed and longevity of the stem cell pool. The work used mouse models to track how NDRG1 levels change with age and injury response. Removing or lowering NDRG1 restored faster repair but reduced long-term cell persistence. The finding points to a need for interventions that selectively modulate NDRG1 rather than eliminate it. Next steps include testing whether partial NDRG1 inhibition can improve healing without compromising stem cell reserves in older animals. A few episodes back, we examined cellular aging markers. This work identifies NDRG1 as a concrete example of the repair versus survival balance. From a molecular brake on repair, we move to a larger-scale vascular mechanism behind common strokes. Researchers re-examined pathology data and found that enlarged and damaged small blood vessels deep in the brain correlate more strongly with a frequent stroke subtype than arterial plaque buildup. Standard antiplatelet treatments such as aspirin show limited benefit because they target plaque-related mechanisms. The analysis points to vessel wall integrity and microvascular remodeling as primary drivers. This reframes prevention strategies toward protecting small vessel health through blood pressure control and other vascular measures. Follow-up work will test therapies aimed directly at microvascular repair. Right now, as you sit reading, the small penetrating arteries deep inside your brain are under constant pressure from everyday blood pressure fluctuations. When those vessels enlarge, weaken, or develop microleaks, they can rupture or occlude even without large vessel plaque. Roughly half of all ischemic strokes involve these small vessels rather than the major arteries visible on standard angiograms. The practical takeaway is that consistent blood pressure management and vascular health habits protect the brain's smallest vessels more directly than plaque-focused treatments alone. Both stories underscore how aging tissues balance repair speed against long-term stability. Immune system aging now offers a framework for measuring such trade-offs. Authors in Nature Medicine outlined criteria for selecting immune markers that can serve as endpoints in trials targeting aging biology. The framework emphasizes reproducibility, responsiveness to intervention, and linkage to clinical outcomes. It includes a roadmap for moving candidate markers from discovery cohorts into interventional studies. The guidance addresses gaps in current biomarker validation for immune system aging. Planned next steps involve applying the criteria to existing datasets from longevity-focused trials. While researchers refine what to measure in humans, modeling shows modest dietary shifts already deliver measurable environmental gains without added cost. A modeling study in Nature Food examined dietary shifts in Scotland using vegetables, beans, and eggs to replace portions of processed and unprocessed meat and dairy. The changes lowered greenhouse gas emissions while maintaining nutritional quality and total food expenditure. Researchers ran scenarios that kept overall calorie intake stable and tracked impacts on health markers and environmental metrics. The results showed measurable reductions in emissions alongside stable or improved nutrient profiles. The study highlights that small, targeted substitutions can align climate and nutrition goals without economic penalties for households. From human dietary patterns, we turn to an unexpected cognitive capacity in another species. Bees solved a classic puzzle box task by discovering an untaught sequence of actions to access sugar solution. The insects had no prior demonstration yet developed the method through trial and interaction with the apparatus. The result adds to evidence that bumblebee cognition supports flexible problem-solving beyond simple associative learning. Researchers documented the novel behavior across multiple colonies to rule out individual idiosyncrasies. Further tests will explore how widely this capacity appears across bee species and task complexities. An atypical gravitational wave detection has prompted renewed examination of whether primordial black holes could account for dark matter. The signal characteristics differ from those produced by stellar mass black hole mergers observed previously. Confirmation would require additional events with matching properties and improved waveform modeling. The finding revives a long-standing theoretical candidate that had fallen out of favor. Next observations from upgraded detectors will determine whether the signal repeats or represents a statistical outlier. Surveys and performance measures revealed that students often choose music to sustain attention during study sessions, yet objective outcomes vary with music type and task demands. Some genres or volumes improved persistence on repetitive work while impairing performance on complex material. The study tracked self-reported motivation alongside accuracy and completion speed across controlled sessions. Results indicate that individual preference does not reliably predict cognitive benefit. Recommendations center on matching music characteristics to specific study activities rather than assuming uniform advantage. Sampling of cats and their fleas in the region detected Rickettsia typhi and related pathogens at rates sufficient to sustain human transmission cycles. The work maps local flea populations and infection prevalence to identify high-risk zones. Researchers note that domestic cats serve as amplifying hosts without showing clinical signs. Public health follow-up will focus on flea control measures and awareness in affected communities. Ongoing surveillance aims to track whether prevalence changes with seasonal or land use factors. Decades of field and lab work at Hokkaido University documented that hantaviruses have adapted to specific rodent species, enabling persistent infection without overt disease. The Hokkaido virus strain was among those characterized during this research. Viral replication remains controlled in the natural host while causing severe outcomes upon spillover to humans. The findings clarify why rodent populations can carry the viruses silently across generations. Continued ecological monitoring will track how habitat changes influence spillover risk. The EVN method integrates transcriptomic data and protein sequence conservation to annotate genomes more precisely than prior pipelines. Benchmarking showed gains in both exon-level accuracy and computational efficiency across multiple species. The approach reduces reliance on purely ab initio predictions that often miss or misplace genes. Developers released the software for community use alongside validation datasets. Future updates will extend compatibility to emerging long-read sequencing platforms. Researchers developed enzyme-mimicking nanoparticles that report on their own intracellular journeys without requiring genetic modification of the host cells. The system tracks uptake, endosomal escape, and degradation pathways through catalytic signals. Experiments in multiple cell types revealed consistent patterns of protein corona formation that influence transport. The method offers a scalable alternative to fluorescent tagging for high-throughput screening. Next applications target optimization of delivery vehicles for therapeutic cargo. University of Michigan researchers demonstrated these secondary applications across multi-year datasets. The passive sensing approach provides frequent global coverage at low additional cost. Operational agencies are now incorporating the derived products into environmental monitoring systems. Continued analysis will refine retrieval algorithms for new surface parameters. University of Lausanne modelers used ice flow physics to trace the transport of large erratics across the Swiss Alps during the last glacial maximum. The simulation matches field observations of boulder locations and lithologies. It quantifies how ice dynamics distributed millions of rocks that now shape local landscapes. Validation against independent dating methods supports the modeled timelines. The framework can be adapted to other formerly glaciated regions. A study in organizational behavior and human decision processes introduced time extension self-efficacy as a mediator between chronotype and willingness to assist colleagues outside standard hours. Employees who believed they could shift their sleep schedules showed different patterns of cooperation depending on their natural morning or evening preference. Survey and behavioral data link these beliefs to actual helping incidents. The findings suggest that schedule flexibility policies may interact with individual circadian traits. Follow-up research will test interventions that build time extension confidence. Before we go, keep an eye on follow-up NDRG1 modulation tests in older mammals that will clarify whether selective changes improve healing without depleting stem cell reserves. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at NARA Network. Links in the show notes. And before you go, this show is part of the NARA Network, a family of daily podcasts on tech, science, and markets, each one a short, sharp briefing you can finish on the commute. If today earned its place in your feed, here's your next listen. Environmental Intelligence, the Environment and Climate Policy Brief for Canada. Find every show free at naranetwork.com. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.